All right, so today we're going to look at cell cycle. So this is a nice illustration in your text showing the cell cycle. The yellow are your mitotic spindles. The little sunburst in the middle are your centrioles. The blue circles are your DNA. And so you can see as you start to enter into cell cycle, your DNA condenses from chromatin into chromosomes. And then when they get ready to divide, they are fully condensed. I like to look at them as little worms. They line up in the middle to be separated. And then that mitotic spindle pulls the cells to opposite poles of the cells. And then you get new daughter cells formed from those original cells. This is actually an illustration of meiosis, which creates 4 daughter cells, but today we will be talking only about mitosis. So what is the function of cell division? It can be in our simpler organisms, our prokaryotic organisms, can be asexual reproduction. Same thing with protists, an amoeba here, undergoing asexual reproduction. Can be for growth and development. So to make new cells so you can grow and develop from an embryo up into a full-sized adult. And for tissue renewal, which really means repair. So when you damage your tissues, you stimulate cell division to repair and renew those tissues. So the cell cycle is broken into different phases, and within those phases you have subphases. So where the cell spends the majority of its time is in this phase called interphase. So 90%, so you can see that 90% of our time in cell cycle is spent in interphase. And then interphase is further broken into G1, S phase, or DNA synthesis, and G2. And the G stands for gap. Gap 1, gap 2, and our DNA synthesis. This first phase, G1, is where you start getting replication of organelles and things that you're going to need to make 2 cells. So you have to make more of what each cell needs so that you can make 2 complete cells. During S phase, this is where we replicate the DNA. And so your homologous chromosomes, those 46 chromosomes that you have, will be replicated, and for a short time your cell will contain 92 of the chromatids that make up the chromosomes. And then in G2 it does a final prep, a final check, finishes making more plasma membrane, cytosol, all those things that you're going to need to make 2 complete cells. Once it finishes interphase, so during interphase, we are preparing. We're making more organelles. We're making more plasma membrane. We're making more cytosol. We're duplicating our DNA so that each new cell can have a complete copy of the chromosomes that the original cell had. And then once we're ready, we move into the next phase, our mitotic phase. And the mitotic phase is made up of mitosis, which is really nuclear division. So we're going to separate the chromosomes, divide the chromosomes between the 2 cells. And then cytokinesis, which is going to divide the actual cell, pinch it in half and make 2 new daughter cells. So make sure you realize you have interphase, mitosis, cytokinesis is the cell cycle. Interphase, where you spend 90% of your time, is G1, S, and G2. And mitosis, we'll see, is divided into 4 phases that I like to tell everybody to remember: PMAT, prophase, metaphase, anaphase, and telophase. And so one thing to note when we are replicating our DNA, so our DNA needs to be in the form of chromatin when it is being replicated, so it is not condensed. Each cell in your body has 23 chromosomes that are each made up of a homolog, so that's a total of 46. You got one from dad, one from mom. When the DNA is replicated, each of the homologous chromosomes, the one you got from mom, the one you got from dad, is replicated exactly. And when they are replicated exactly, they are called sister chromatids. So our homologs are going to contain the same genes, and genes are what code for proteins. But one may code for brown hair, and one may code for blue hair. Yeah, we've seen somebody born with blue hair, haven't we? But brown eyes, brown hair, blonde hair. So they contain the same genes that are going to code for traits within an organism, but they may be different versions of those genes. And so homologous chromosomes contain the same information for what an organism is going to be, but it may be slightly different versions of that. But your sister chromatids are identical copies. They are identical to each other. And so homologous chromosomes are similar but not exact, whereas our sister chromatids are exact. And then when we separate at the end of mitosis, one of each sister chromatid ends up in our new cells. So that each new cell, each new daughter cell, has a complete set. So it has the entire set of homologous chromosomes, the 23 from dad and the 23 from mom. Just make sure you don't confuse the difference between homologous chromosomes and sister chromatids. And so here's another depiction of how many chromosomes a human cell has. So again, eukaryotic chromosomes are made up of chromatin. Chromatin is where the DNA is replicated when it's in this form. When the chromatin condenses into tightly packed rosettes of DNA is when it becomes chromosomes. A somatic cell has 46 chromosomes, so 2 sets of 23. And that one set from mom, one set from dad. And so here you can see before interphase, 46, 46. After interphase, you still have just 46 chromosomes, but you have those identical copies of those 46, so you have a total of 92. And then at the end, those 46 will be equally divided between the two daughter cells. But you're going to get an equal division of the sister chromatids into each cell. So this cell now has a complete set of homologs, and this one has a complete set of homologs. And here is what a highly condensed chromosome looks like. And so these are the sister chromatids held together by a centromere. And so this is where the mitotic spindle is going to connect. It connects to the kinetochore. And so this is one sister chromatid, this is one sister chromatid, identical copies of each other. And here, as we move through the phases of mitosis, prophase, metaphase, anaphase, you can see how the chromosomes become even more condensed and that you have this separation. You can start seeing here in anaphase the separation of the cohesion fibers that hold the two sister chromatids together, where they'll be pulled to opposite poles, the sister chromatids. So let's look at the phases of mitosis. So remember, we went over interphase. Interphase was G1, S, G2. Purpose of interphase is to replicate organelles, increase cytoplasm, increase your plasma membrane, so you have enough plasma membrane to form two new cells, getting everything ready so that you have enough materials to replicate. And so here, the blue is our nucleus, and you can see how it's a very evenly homologous, dispersed blue circle. And that's because the chromosomes are duplicated, but they are uncondensed, so they're in that form of chromatin. You can see the mitotic spindle, the centrioles here, getting everything ready, and then all this red would be your cytoskeletal elements out there. So we enter into mitosis. The first phase of mitosis is prophase, and so a lot of books like to break this into prophase and prometaphase. But for our purposes, you can just combine these into prophase. And so you can see we're starting to get little worms and really have all the worms by the end of prophase. And so this is when this chromatin, the stringy ball of material, has fully condensed into these tightly packed chromosomes. And so they are condensed. So that is one of the things that happens during prophase, is that the chromosomes condense. You start to break down the nuclear envelope. So the nuclear envelope is broken down and the mitotic spindle forms. So the centrioles move to opposite poles and start putting out these microtubule fibers to form the mitotic spindle. And the mitotic spindle will start hooking to those sister chromatids at the area called the kinetochore. So the mitotic spindle wiggles and jiggles and pushes those sister chromatids to the middle of the cell during metaphase. So all of our homologous chromosomes are lined up, a sister chromatid facing each pole of the cell. So the homologs are lined up, sister chromatid on each side of the mitotic plate, and a mitotic spindle hooked to each kinetochore of each sister chromatid. And so during metaphase, the chromosomes all line up at the mitotic plate. Now during anaphase, the cohesion proteins have been broken down. The sister chromatids are no longer held together. And so during anaphase, the sister chromatids are each pulled to the opposite pole of the cell by our mitotic spindles. And then at the end, when we get to telophase, telophase is just the reverse of prophase. So our chromosomes start to uncondense. Their nuclear envelope starts to reform. The mitotic spindle breaks down. And then after telophase, it is followed by cytokinesis, which ends up cleaving the cell into two new daughter cells. And so as you look, here is the actual photomicrographs of this. You can see the mitotic spindle is the yellow, the chromosomes are the blue worms, and you can see all the cytoskeletal elements in red, so the outline of the cell. And the mitotic spindles have pushed and lined up all of our chromosomes in the middle. When we get to anaphase, the mitotic spindle starts to degrade and pull the sister chromatids to opposite poles. And then in telophase, we start to form two new daughter cells. The DNA starts to uncondense, and your nuclear envelope starts to reform. So here is a real cool video of this. In real time.
In this time-lapse video of cell division in a newt lung cell, you can see a process that normally takes more than an hour occur in less than a minute. This newt cell has already started prophase, the first stage of mitosis. Chromosomes have condensed and become clearly visible, contained within the nuclear envelope. In the cytoplasm, the mitotic spindle begins to form during prophase. During prometaphase, the nuclear envelope breaks up. The chromosomes become even shorter and thicker. The microtubules of the mitotic spindle attach to the chromosomes and move them toward the equator of the cell. In metaphase, the chromosomes compete at the metaphase equator, imaginary important and distant points in their poles. Anaphase begins when the paired centromeres age chromosomes separate, and the sister chromatids, now full-fledged chromosomes, are moved to the pole. The walls of the cell are moved to parts to the cell. During telophase, two darker nuclei form, and the chromosomes become less tightly connected. Cytokinesis begins during telophase, resulting in two identical chromosomes.
So that's a real cool thing to see, is the actual cell division. And you can find there's lots of really good videos, remember, in my study area, and you can look under study by chapter and view a lot of these videos. There's a lot of videos associated with the chapter, so go check it out. And I want just to show you, this is an experiment that was run. So are the spindles retracting from the spindle side, or are they retracting from the chromosome side? And so the experiment was, these guys took a laser and they marked the microtubules, the mitotic spindle, and then they looked at which side shortened from where they'd marked it with the laser. And what they saw is that the side connected to the centromere of the sister chromatid is what was actually shortening to pull the chromosomes to the opposite poles. And what actually happens is that you have this motor protein, like a little Pac-Man, wonk, 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 chewing up that microtubule and spitting out those tubulin subunits, which will probably be reused to make more mitotic spindle at another time. And so it's basically degrading that mitotic spindle, which is a microtubule, and shortening it to pull those sister chromatids to the opposite poles. There's another illustration, and you can see that again under my study area, so be sure to go check out our videos in my study area. There is a unique difference between animal cells and plant cells. So animal cells have a cleavage furrow that is made by a contractile ring of microfilaments, which pinches it in half. But we know plants have a cell wall, and so they have to have an additional or a different type of cytokinesis where vesicles form the cell plate, which eventually fuse to form a complete new cell wall between the two new daughter cells in a plant cell. And so this is the final phase with prophase. So this is an onion root tip. Prophase, very diffuse DNA. You can see the cell wall. Here with prophase, you see the chromosomes starting to condense. You can't really see the nuclear envelope breaking down or the mitotic spindle, but you can definitely see the chromosomes condense. During metaphase, they're lined up in the middle. Again, you can see the outline of the cell wall. During anaphase, they are pulled to opposite poles. This is not the best one of anaphase, but they start to get pulled to opposite poles. And then in the final step of telophase, cytokinesis, you start to see the chromosomes uncondense, nuclear envelope reforms, and you see the formation of a new cell plate between the two new daughter cells. All right, so this is cell cycle. I hope you enjoyed it. Tune in for the next one on cell cycle control.